Hello, folks. Welcome to part two of the 1.2 topic in Advanced Higher on proteins. We did the first half in the last video, and we're going to just resume the topic and work our way through to the end of it in this video. So we're going to kick off by looking at protein structure, ligand binding, and conformational change. So from higher, you probably remember, and even from that 5, you'll probably remember that proteins are made of amino acids, okay? And to use a couple of chemistry terms to explain that, we would say that proteins are polymers, poly meaning many, of amino acid monomers, mono meaning one, okay? So amino acids are the individual units which make up proteins. Now, amino acids are linked by peptide bonds to form polypeptides. Again, we probably know that from higher already. One thing you have to be able to do at advanced higher is you have to be able to recognize the chemical structure of a peptide bond. And there's an example in the diagram down here where we can see a peptide bond there, okay? And what we can see is that we've got a condensation reaction happens. Again, to talk a little bit of chemistry, you don't need to know this for the exam, but so that from one side here and the H from this side are released in a condensation reaction, and the carbon and the nitrogen there form a bond. Okay, so we're looking for the loss of the here and the H here and a bond between the carbon and nitrogen for the peptide bond. Other ways to look at it are you look for your amine group over here at this side. You should have your carboxyl there, but the OH is missing. Similarly, you've got your full carboxyl group there, and you've got the H missing from the amine group there. So that is how you can spot a peptide bond, okay? And that's something you have to be able to do for the exam in a diagram. Amino acids have the same basic structure, differing only in the R group that's present, okay? So again, we can see our amine group there, we can see our carboxyl group here, and our hydrogen here, okay? And then the R group is whatever is opposite that hydrogen, okay? Now, just be conscious that sometimes the R groups can be carboxyl or amine, and so you have to make sure that you look at the structure properly and you recognize the carboxyl and amine being across from each other, and the hydrogen is across from the R group there. Okay, so amino acids are classified according to their R groups. There are 4 groups we need to know about. The basic group, which are positively charged, and the things to look out for in a basic group are NH2, NH3 structures. We've got the acidic group, which are negatively charged, and we're looking out for the carboxyl there, okay, the COOH. We've got a polar R group, which are hydrophilic, so they are loving water, and that's because they have an okay, oh my gosh, there's so much water, and hydrophobic. And again, when we're looking at the hydrophobic one, hydrophobic ones don't like the water. We tend to find them deep within the protein. And to recognize those in terms of their structure, we're looking out for these hydrocarbon chains, okay? If you see an amino acid and it's got a large number of hydrocarbon chains, it's probably hydrophobic R group. Okay, so the R groups of amino acids can vary in a number of ways, through size, through shape, through charge, through hydrogen bonding capacity, and also chemical reactivity. And we can tell that just by thinking about the variety of the R groups that we have here. You can see the variation in charge and the likely variation in shape that comes from obviously having a different arrangement of molecules in there and atoms in there, and hydrogen bonding capacity similarly, and chemical reactivity. So all of these things, the variations will cause and structure will cause variations in all of these other areas as well. Here are some examples of the various groups. So we've got our hydrophobic ones here, we've got our basic ones here, we've got our acidic ones here, and our polar ones here. Okay, you don't need to memorize these or even know the names of all of the amino acids, but you do have to be able to recognize them if you see them in a diagram in the exam. So it's worth just reviewing, maybe pressing pause in this video and reviewing this diagram here, and making sure that you could recognize each of the R groups if they were presented to you. Okay, and you can see one of the good things about this diagram is you can see the common structure of the amino acid, the common features, the amine, carboxyl, and the hydrogen there, and you can see that in all of them. And hopefully, again, that will help with your familiarity of recognizing the structure of the R groups. A couple of good videos that I found on YouTube for this one as well, which you can watch. Again, we're not going to watch a video within a video, but you can look these up and watch them yourself. So there's this one, which is quite good, and this one here, which is excellent at linking what we've just learned in terms of the R groups to the next thing, which is all about protein structure. So the wide range of functions carried out by proteins results from the diversity of our groups. Okay, by having such a wide range of them enables us to combine and form different shapes with our polypeptide chains and therefore produce different shaped proteins. And as we know, the variety of shapes of proteins gives them a variety of functions. Now, when we're talking about the structure of a protein, we talk about four different structures. We talk about first of all, the primary structure. Now, the primary structure is simply the sequence of the amino acids, okay, and the order or the sequence in which they're synthesized into our polypeptide during translation at the ribosome. Okay, so that initial sequence of polypeptide is the primary structure. Now, we have in a secondary structure, we have hydrogen bonding. We have hydrogen bonding throughout our proteins, but we have hydrogen bonding along the backbone, and that results in these regions here called alpha helices. You can see an alpha helix in this diagram over here. Okay, there's our alpha helix, and we get parallel and anti-parallel beta-plated sheets. Okay, and we can see that. We can see it going one way and then the other way. So we get the anti-parallel there, one way, then the other way, and we get the parallel one on the right here. We also have turns, and we can see the turns on the sheets there. That's a turn, 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 another turn here on the helix as well. And these are the things that you need to look out for when you're trying to identify a secondary structure. Quite often in the exam, they'll show you part of a protein at some stage of its development, and you have to identify if it's in the primary structure, the secondary structure, the tertiary, or the quaternary structure, which we're going to have a look at in a minute. Okay, so these are the features to look out for in the secondary structure. The peptide will continue to fold until it eventually achieves a tertiary structure, and we can see again what a tertiary structure looks like in these diagrams at the top here. Okay. And that conformation, that shape that the protein takes, that is stabilized by a number of things, okay, which include interactions between our R groups, okay. So, for example, our basic and acidic will interact with each other to form ionic bonds. We've also got hydrophobic interactions, again, on the basis of our hydrophobic R groups. London dispersion forces, the chemists will know lots about that. Hydrogen bonds, which we've already mentioned before. Disulfide bridges, which provide a covalent bond between R groups that contain sulfur. Okay, so that's the tertiary structure held together by all of these bonds and interactions. So you need to know that list for your exam, and you have to be able to list those if requested. The quaternary structure exists in proteins with two or more connected subunits. Now, those subunits are long chains of polypeptide which have been folded and are already in their tertiary structure, and then they're joined together with other subunits to form the quaternary structure. We use the term quaternary structure to describe the spatial arrangement of those subunits. Now, sometimes within a series of subunits like this, we have something that's known as a prosthetic group, okay? Well worth remembering that term because it comes up a wee bit. And a prosthetic group is a non-protein unit, and it's tightly bound to a protein and necessary for its function. Probably the most common example of that is the heme group, which is part of the hemoglobin structure. And the heme is necessary. Okay, heme is not protein, but it's attached to the hemoglobin, and it's obviously an essential part of the function of hemoglobin, which obviously is to bind oxygen to that heme group. Okay, the ability of hemoglobin to do that is severely compromised if it does not have a heme group. Okay, so that non-protein, that prosthetic group, allows it to do that function. Okay, factors which affect the R groups within proteins. There are two main ones, and you're probably familiar with these from your previous study of proteins, and they are temperature and pH. Now, if we increase the temperature, it's going to disrupt the interactions that hold the protein in shape. The protein will begin to unfold and eventually become denatured, okay? And we know from our previous study that denatured means for the protein to lose shape, okay? And that's how it happens. We disrupt the interactions and it begins to unfold. Now, the charges on the acidic and the basic R groups are affected by pH, okay? As the pH either increases or decreases from the optimum, the normal ionic interactions between those charged groups, between the acidic and basic charged groups, are lost. And that gradually changes the conformation of the protein until it becomes denatured, okay? Conformation is another word for shape, okay? So the charges in the acidic and basic R groups are affected by pH. And because we will either increase or decrease the pH, that will disrupt those ionic interactions and that will gradually change the conformation of the protein until it becomes like this diagram on the right, unwound and denatured. Okay, it's lost its conformation. Ligands or ligands, however you want to say it. So ligand binding changes the conformation of a protein. So what is a ligand? Well, a ligand's any substance which binds to a protein. In your previous studies, you'll have learned about substrates and enzymes. The substrate is an example of a ligand because it binds to the enzyme, which is a protein. Okay, so anything that binds to a protein is a ligand. R groups not involved in the protein folding, okay, in the three-dimensional shape that arises from all the initial folding. Those R groups can allow binding to ligands, okay? So the R groups that are not involved in protein folding are involved in binding to ligands. And what they have is they have these binding sites, okay? You'll remember we talked about an active site on enzymes. And binding sites have a complementary shape and chemistry to the ligand, okay? Again, some of this, like the concept of this is probably familiar to you from your previous study of enzymes. So we're just applying it to all proteins and all substances which bind to a protein. Okay, so those binding sites have a complementary shape and chemistry to the ligand. Now, as a ligand binds to a protein binding site, the conformation of that protein will change. Similar to when our substrate bound to our enzyme, we had that induced fit model where the shape of the active site would change to better fit our substrate. Any binding of a ligand to a protein will cause some kind of conformational or shape change within that protein. And of course, with proteins, we know that shape follows function. So if we change the shape, the following function will also be altered. Okay, so there'll be a functional change in the protein as a result of that conformation change. Allosteric molecules. So allosteric interactions occur between spatially distinct sites, okay, i.e. the 4 subunits here that make up the quaternary structure of this molecule. Many allosteric proteins contain multiple subunits, okay, i.e. have quaternary structure. Now, allosteric proteins with multiple subunits tend to show something called cooperativity. Now, cooperativity is where if there is a change in the binding at one subunit, it will alter the affinity of the remaining subunits. And a good example of that is something like hemoglobin and oxygen. So when oxygen binds to one of the heme groups, that then increases the affinity for oxygen on all of the other heme groups as well. So what you do is you get very, very quick changes in the oxygen saturation due to small changes in oxygen within the person's blood. Okay. So the binding of the substrate molecule to one active site of an allosteric enzyme increases the affinity of the other active sites for binding to subsequent substrate molecules. Okay. So that's just a general definition of the example I gave, the specific example I gave of oxygen and hemoglobin. Why is this of biological importance? Well, the activity of the allosteric enzymes can vary greatly with just small changes in the substrate. Okay, so just small changes in the oxygen concentration will have a big impact on the activity of the hemoglobin molecules in a red blood cell. Okay, they will suddenly have a high affinity when oxygen concentration is a bit higher, and then without much notice, they will very quickly go down to having quite a low affinity as the oxygen concentration drops, even just slightly. And we see that drop off when in hospitals when people's oxygen saturation levels they go below sort of 90 85% or so. When they dip below that, we suddenly get this decrease in oxygen saturation, whereas it's quite it's quite a stable saturation at above 90 or so. Okay, allosteric enzymes contain a secondary site. This is called an allosteric site. Now, you'll be familiar with the concept of this again from your look at non-competitive inhibitors at higher, okay? Non-competitive inhibitors bound away from the active site, In advanced higher, we're given that that site a name, and we're saying it's called an allosteric site, okay? And we can see it on this diagram here, the allosteric site, which is away from the active site. Now, modulators, they are molecules which regulate the activity of the enzyme when they bind to the allosteric site, okay? So, like non-competitive inhibitors are an example of a modulator. So, following binding of the modulator, the conformation of the enzyme changes, and this alters the affinity of the active site for the substrate. And again, we saw that with non-competitive inhibitors. They bound away from the active site, but their result was that they changed the active site's shape, and therefore the substrate could no longer bind. Okay, it reduced the affinity for that substrate. Positive modulators are those that increase the enzyme's affinity. Okay, they increase the enzyme's affinity, whereas negative modulators, they reduce. Okay, so there are two types of modulators you'll need to know about for your exam. The positive ones increase the affinity, the negative ones inhibit or reduce the affinity of the enzyme to the substrate. The binding and release of oxygen hemoglobin shows this cooperativity, okay, and it shows these changes that we discussed earlier on. Okay, the influence and physiological importance of temperature and pH on the binding of oxygen. So changes in binding of oxygen at one subunit alter the affinity of the remaining subunits for oxygen. We talked about that example in the previous slide. So a decrease in pH or an increase in temperature, they will lower the affinity of hemoglobin for oxygen. So the binding of oxygen therefore becomes reduced. Okay. So a decrease in pH will lower the affinity of hemoglobin for oxygen. So therefore the binding of oxygen is reduced. Okay. And we can see that in our graph here. As the pH goes down, we can see that reduction there in oxygen hemoglobin attachment. Reduced pH and increased temperature in actively respiring tissue, okay, so when we're doing exercise, will reduce the binding of oxygen to hemoglobin, okay, it will reduce the binding of the oxygen to hemoglobin, and that in turn will promote increased oxygen delivery to our tissues. And that makes sense, doesn't it? Because when you are exercising intensely, you're going to reduce the pH because you'll be producing lactic acid. You'll have an increased temperature because your body will be increasing its metabolism and the amount of heat generated will increase. Therefore, it makes sense that that would cause a lowering of the affinity for oxygen to hemoglobin. And therefore, that's good for us because it promotes an increased oxygen delivery to our tissues. We'll get more oxygen into our tissues, which we know we need for aerobic respiration within the mitochondria of the cells to produce the ATP that we require for the muscle contraction that is happening during our intense exercise that we're doing. So we're going to talk about two enzymes that are responsible for causing conformation changes within proteins. Now, the addition or removal of phosphate. Now, we learned about phosphorylation and higher. Okay, so the addition or removal of phosphate, that's phosphorylation to add the phosphate to the protein. That can cause reversible conformational change in proteins, okay? We can temporarily or reversibly cause a conformation change in the protein by either adding or removing a phosphate molecule. And we can see that in the diagram over here on the right. Okay, we can see the phosphate been added to this protein. Okay, now this is a form of post-translational modification. Anything that happens after the protein has left the ribosome is going to be an example of post-translational modification. So the addition of this phosphate will come at a later point in the protein's development. Sometimes it's used to activate or inactivate the protein, okay, depending on whether we need it active or deactivated at certain places within the cell in the body. Now, we need to know about two different types of enzymes involved here. We've got protein kinases. They catalyze the transfer of a phosphate group to another protein. And we can see kinase in action here. It's removing the terminal phosphate from ATP, the third of the adenosine triphosphate, okay, the third phosphate from this molecule. And it's breaking that down into ADP plus phosphate. And the phosphate is then added to our protein here via this enzyme called protein kinase. As I said before, it's the terminal phosphate of the ATP that is transferred to the R groups, okay, of the protein. Now, protein phosphatases catalyze the reverse reaction, okay? So that'll be taking the phosphate from here, and they will then take that phosphate away. That phosphate will then have the ability to bind with ADP to form ATP again, and we'll get that regeneration of ATP that we need to have constantly cycling within our cells. So phosphorylation brings about conformational changes. That's the key thing to remember, and that can affect a protein's activity. Sometimes it will activate it, sometimes it will deactivate it, depending on what the specifics are of that particular protein. Now, the activity of many cellular proteins, such as enzymes and receptors, are regulated in this way. Okay, they're both examples, as we know from Nat5 biology, of proteins. So, some proteins are activated by the phosphorylation, they are activated, while others are inhibited, as we mentioned earlier. Now, this is an important point. Adding a phosphate group adds a negative charge to the protein. And what this means is that ionic interactions in the unphosphorylated protein can be disrupted, and new ones created. Okay, so adding that extra negative charge from the phosphate group will affect the ionic interactions within the unphosphorylated protein. So you know that those basic and acidic R groups, which are interacting with each other ionically, they are going to be interrupted by the addition of that phosphate. So that's worth considering and remembering as well. Okay, folks, so that's the end of topic 1.2 on proteins. That's the second part of 1.2. It was a long old topic. We're going to be back next time for 1.3. But until then, leave any comments. Let me know if you enjoyed anything in the video, if you have any questions, if there's anything you're still not sure about. But until the next video, thanks for joining me, folks, and class dismissed.